Okay, Chapter 3, Review, PowerPoint. Chapter 3 is all about organic compounds. Organic compounds are those that have carbon-based compounds, basically. So lots of carbon bonded to itself in chains, branches, and rings. So a compound containing carbon is said to be an organic compound. We also call these macromolecules. So we have 4 different classes of organic molecules: carbohydrates, your sugars; lipids, your fats, oils, and waxes; proteins; and nucleic acids, DNA and RNA. Carbon is very versatile. The reason why carbon is very versatile is because of its 4 valence electrons, which allows it to basically bond to itself and other atoms in 4 directions. With 4 valence electrons, carbon can form 4 covalent bonds with a variety of atoms. This ability makes large, complex molecules possible. In molecules with multiple carbons, each carbon bonded to 4 other atoms has a tetrahedral shape. However, when 2 carbon atoms are joined by a double bond, the atoms joined to the carbons are in the same plane as the carbons. When a carbon atom forms 4 single covalent bonds, the bonds angle toward the corners of an imaginary tetrahedron. When 2 carbon atoms are joined by a double bond, the atoms joined to those carbons are in the same plane as the carbons. So this just looks at different shapes of carbon-based compounds. The electron configuration of carbon gives it covalent compatibility with many different elements. The valences of carbon and its most frequent partners, hydrogen, oxygen, and nitrogen, are the building code that governs the architecture of living molecules. So hydrogen's valence is 1 because it needs 1 electron to fill its outer shell. Oxygen has 6 valence electrons, so it only needs 2 more to fill its shell to make 8. Nitrogen has 5, so it needs 3. Carbon has 4, so it needs 4. Carbon atoms can partner with atoms other than hydrogen, for example, carbon dioxide or urea. Here are 2 sex hormones. Carbon chains form the skeletons of most organic molecules. They can vary in length and shape. So here are some hydrocarbons, those molecules that have just carbon and hydrogen as their elements. Hydrocarbons are organic molecules consisting of only carbon and hydrogen. Many organic molecules, such as fats, have hydrocarbon components. Hydrocarbons can undergo reactions that release a large amount of energy. Functional groups. These are the molecules that are most commonly involved in chemical reactions. The number and arrangement of functional groups give each molecule its unique properties. So here are your functional groups: hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, phosphate, and methyl. Okay, so hydroxyl, OH, makes a molecule in alcohol, like ethanol. For carbonyl groups, C double bonded to an O, we can have a ketone, which would be a carbonyl group in the middle of a molecule, or an aldehyde, found at the end of a molecule. Carboxyl group, C double bonded to an O, bonded to an OH, such as in your amino acids, for example. Amino group, another functional group in an amino acid, so just NH2. Sulfhydryl, an S and an H. Phosphate group, P with 4 O's, and methyl group, C with 3 H's. One organic phosphate molecule, adenosine triphosphate, ATP, is the primary energy transferring molecule in the cell. ATP consists of an organic molecule called adenosine attached to a string of 3 phosphate groups. So here's ATP. ATP can store an incredible amount of energy because ATP is a very highly unstable molecule because of these negative charges that repel. So when ATP reacts with water, an inorganic phosphate comes off of the molecule, and a great amount of energy is released because ADP, adenosine diphosphate, is much more stable than adenosine triphosphate. Okay, now we're looking at polymers. Polymer is a large molecule consisting of many similar building blocks. These building blocks are called monomers. Some molecules that serve as monomers also have other functions of their own. Okay, synthesis and breakdown of polymers. We have dehydration reactions where water gets taken out. So let's look at that here. So here we have a dehydration reaction. We have a hydrogen being taken off of one molecule, and an OH, a hydroxyl group, being taken off of another. So as a result, that makes water. That's the dehydration part. And then remember, this is also called synthesis. Dehydration, synthesis. We're taking out water and we're making a larger polymer, also called condensation synthesis or condensation reaction. Versus hydrolysis, which is the breakdown of a polymer where water is getting added and it's breaking apart this polymer. Each cell has thousands of different macromolecules. Macromolecules vary among cells of an organism, vary more within a species, and vary even more between species. An immense variety of polymers can be built from a small set of monomers. So first we have carbohydrates. Carbohydrates are sugars and the polymers of sugars. The simplest carbohydrates are monosaccharides. Glucose would be an example of a monosaccharide. Carbohydrate macromolecules are polysaccharides, polymers composed of many sugar building blocks. So monosaccharides have molecular formulas that usually are multiples of CH2O. Glucose is the most common, C6H12O6. Monosaccharides are classified by the number of carbons in the carbon skeleton and the placement of the carbonyl group. Okay, so here we have a glyceraldehyde, which is a 3-carbon sugar. We have ribose, and then we have glucose versus fructose. Notice the difference is, one of the differences, well, the primary difference is the location of the carbonyl group. In fructose, it's in the interior of the molecule, and for glucose, it's on one end. And then here we have each of them. Though often drawn as linear skeletons, in aqueous solutions, many sugars form rings. Monosaccharides serve as a major fuel for cells and as raw material for building molecules. Okay, so here we have the linear form of glucose, and then when you put it with water, it forms this ring structure. A disaccharide is formed when a dehydration reaction joins two monosaccharides. The covalent bond is called a glycosidic linkage. Okay, so dehydration synthesis, removing OH and an H to form water. And then the linkage between this carbon and this oxygen and that carbon, that's called a glycosidic linkage. So that's unique to carbohydrates. Polysaccharides, the polymers of sugars, have storage and structural roles. The structure and function of a polysaccharide are determined by its sugar monomers and the positions of glycosidic linkages. Starch is the storage polysaccharide of plants. Plants store surplus starch as granules. The simplest form of starch is amylose. Glycogen is the storage form of sugar in animals. Humans and other vertebrates store glycogen mainly in the liver and muscle cells. Here we have starch, glycogen, and lastly cellulose. Cellulose is the major component of the cell walls of plants. Like starch and glycogen, cellulose is a polymer of glucose, but the glycosidic linkages in cellular differ. The difference is based on two ring forms of glucose. Okay, so starch has 1,4 glycosidic linkages of alpha glucose monomers, so notice that basically these glucose molecules are all right side up. Versus the 1,4 linkage, every other glucose molecule is, in a sense, upside down. In starch, glucose monomers are arranged in the alpha conformation. Starch and glycogen are largely helical. In cellulose, the monomers are arranged in the beta conformation. Cellulose molecules are relatively straight. In straight structures, hydrogen atoms on one strand can form hydrogen bonds with OH groups on the other strand. On the other strands. Parallel cellulose molecules held together this way are grouped into microfibrils, which form strong building materials for plants. Enzymes that digest starch by hydrolyzing alpha linkages can't hydrolyze beta linkages. This is why cellulose in human food passes through our digestive tracts as insoluble fiber. Some microbes use enzymes to digest cellulose. Many herbivores, from cows to termites, have symbiotic relationships with these microbes. Chitin is another structural polysaccharide, and it's found in the exoskeletons of arthropods. Chitin also provides structural support for the cell walls of many fungi. Next we have lipids. These are your fats, oils, and waxes. They are hydrophobic because of their many nonpolar covalent bonds. They have little or no affinity for water as a result. The most biologically important lipids are fats, phospholipids, and steroids. Fats are constructed from two types of smaller molecules, glycerol and fatty acids. Glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon. A fatty acid consists of a carboxyl group attached to a long carbon skeleton. Okay, so here again, dehydration synthesis, you have the removal of an OH group from the fatty acid, the removal of a hydrogen from the glycerol, and then that forms what's called an ester linkage. Okay, so here we have 3 fatty acid chains that are attached to one glycerol molecule. This is a triacylglycerol or a triglyceride. Fats separate from water because water molecules hydrogen bond to each other and exclude the fats. In a fat, 3 fatty acids are joined to glycerol by an ester linkage like you just saw in the previous slide, creating a triacylglycerol or a triglyceride. Fatty acids vary in length and in number and location of double bonds. Saturated fatty acids have the maximum number of hydrogen atoms possible and therefore no double bonds. Unsaturated fatty acids have 1 or more double bonds. Okay, so here's a saturated fat, which is solid at room temperature. You can see that all of the fatty acid chains are straight because they do not have any double bonds like you see here. Because there are no double bonds, then there can be tight packing of that fat, which makes it solid at room temperature. Versus your unsaturated fatty acid, we have a double bond here which causes a bend in the molecule. Unsaturated fats are liquid at room temperature as a result. Fats made from saturated fatty acids are called saturated fats and are solid at room temperature. Most animal fats are saturated. Fats made from unsaturated fatty acids, called unsaturated fats or oils, are liquid at room temperature. Plant fats and fish fats are usually unsaturated. Major function of fats is energy storage. Fat is a compact way for animals to carry their energy stores with them. Next we have phospholipids. Phospholipids are found in your cell membranes. They consist of 2 fatty acids and a phosphate group that's attached to glycerol. The 2 fatty acid tails are hydrophobic, and the phosphate group and its attachment form a hydrophilic head. Okay, so here are the fatty acid tails of the phospholipid, and here's the hydrophilic head. Here's your phospholipid bilayer, your cell membrane. The hydrophobic tails point toward each other, and the hydrophilic heads point inside and outside of the cell where there is water. When phospholipids are added to water, they self-assemble into a bilayer, with the hydrophobic tails pointing toward the interior. This feature of phospholipids results in the bilayer arrangement found in cell membranes. Steroids are lipids characterized by a carbon skeleton consisting of 4 fused rings. Cholesterol, an important steroid, is a component in animal cell membranes. So it helps control the fluidity of the cell membrane. And here's cholesterol. Okay, that concludes part 1 of Chapter 3 review.